Okay. I guess it's still break. Okay. Okay. I'll give it a couple of minutes. That's okay. After 12 o'clock, there's nothing until lunch. Okay. And I can see some people already are eating lunch, so there's no rush, right? Now, what I'll do is I'll finish off the MSc, just a tiny portion left, and then move on to immunization lecture. What we talked about was the genetics of the MSc, what the genes and what the loci are, and how the diversity or the polymorphism is generated, for class 1 and class 2 MSc antigens, and how they are inherited, and what is the role of the mixed lymphocyte reaction, in other words, alloantigen recognition, expansion of T cells, proliferation of T cells in response to class 2 MSc antigens, and induction and activation of cytotoxic cells, the cytokines that are generated during the process, and how it can damage the graft very, very quickly. Of course, later on, there is, as the organ releases specific antigens, those are alloantigens and other antigens as well, MSc antigens and other antigens, they are not processed and presented to the host T cells in a normal manner, and there is induction of antibody response that in turn again will cause damage, either complement-mediated that you've already learned from Dr. Mayer, as well as the ADC that both Dr. Mayer and Dr. McCallum have mentioned. So that is where we were. What I'm going to do is, one of the things that I did not tell you about was that, but you saw that, what is the nomenclature of the MSc antigens. And you will see something like A21 or A3, that refers to the polymorphic form. 3 refers to one of the polymorphic forms, and A refers to the locus. And in the case of D, P, DQ, and DR, it is the same nomenclature, the letters followed by a number. That was somewhat older nomenclature. With the molecular biology techniques that they are detecting actually the genes, what they do is it's designated by the letter followed by a 4-digit number. And first 2 digits are actually refer you back to the old nomenclature. So if you see A03 followed by 2 more numbers, A03 actually refers to the old nomenclature. That means the A3, third polymorphic form of A locus antigen. And then there is another 2-digit number that is actually referring to the sub-polymorphic forms. Actually, since they were serologically defined old ones, there are some other polymorphic forms as well within that group, and that is detected by PCR. Okay? So the new nomenclature is a 4-digit number. That's all you have got to bear in mind. And that is based on the PCR-based sequence of the locus. And of course, different individuals would have different sequences. That's what gives it antigenic uniqueness. So that's the system for nomenclature. Now, all right, depending on how the graft is rejected, how quickly, the temporal characteristic, as well as what the mechanisms are, these graft rejections you'll find in the book as well as in the notes are classified into 4 different categories. And they are the hyperacute rejection, which is within minutes. As a matter of fact, some extreme cases are the organ is transplanted planted, the blood vessels are anastomosed, and then within minutes there are signs of rejection, malfunction of the organ, and the actual the look of the organ can tell you that it is in the process of rejection, and that is referred to as hyperacute rejection. And the mechanism there is pre-existing antibodies. That's why it's very important to cross-match, that the recipient does not have any antibodies that can react to the donor's MHC. Otherwise, the antibodies will combine with the tissue, the complement will be activated, and there will be complement-mediated damage, and there will be inflammatory cells at the junction where the organ has been anastomosed and damage that anastomosis and also damage the organ. So that is the hyperacute rejection, as I said, due to preformed antibodies. Then there is accelerated rejection, and that is due to pre-existing cytotoxic T cells. If there are not antibodies, you may have matched for the antibodies, but there may be pre-existing cytotoxic T cells or memory cells, and these are reactivated, of course. These are reactivated, and they produce their cytokines. Cytokines recruit the effector cells, and effector cells damage the organ. And it is within days after transplantation. And then there is a sort of acute rejection that is days to weeks. And that is that the patient did not have, it will occur in patients that do not have any preformed antibodies or presensitized cells. But as the organ, the foreign antigens stimulate the recipient's cells, immune cells, they are going to produce effector cells and effector molecules, namely antibodies, sensitized cells, and then cytotoxic cells. So that's de novo, that's a naive or the primary sensitization process. So it takes longer than what the time that will take for the presensitized cells to get reactivated. It's like a booster response. Remember Dr. Mayer told you an amnestic or secondary response in a context with antibodies. Well, there is secondary response to graft rejection as well. And that was the accelerated, but the primary response is seen in the acute response, acute rejection. And it is a lot longer than the accelerated rejection. And finally, there is chronic rejection. And that is, the mechanism is not really well known. It is multifactorial, really. The time frame here is months. The patient is okay, organ is functioning all right, and months later, there is a rejection episode. And this may be due to either cross-reactive antigens that have sensitized the individual because of certain infections, or it may be the damage to the organ which was responsible for dysfunction of that organ in the first place. Okay? So there is also believed that organ as it exists in the body, because there's a large amounts of, it's a large tissue and large amounts of antigens that his immune system is exposed, and there is some induction of tolerance to the foreign antigens. And we'll talk about that later in the next not next lecture, but one after that, when we talk about tolerance. So the organ can actually induce tolerance in the body. And for some reason, that tolerance breaks down. Okay, you had a...
In hyperactive reaction, what's the chances of the recipient has antibody already to that MHC?
There are lots of chances. Well, in the hyperacute, the question is, you heard that, what are the chances of the recipient having antibodies? Well, multiple transfusions. Okay? In the case of women, multiparity. Okay? There are infectious agents, pathogens that can induce antibodies that can cross-react with the tissues and organs. And that is the reason it is always done that there is a cross-match between the donor and the recipient. The recipient is always checked for antibodies against the tissue antigens of the donor. Okay? Any other questions? So those are the 4 different modes of rejection that have been defined. Okay, what is most important is, of course, the health of the recipient as well as donor. Donors are both checked for, the recipient is checked for hypertension and any other routine, examination, physical examination. He should be in reasonable health. But after that, the cross-matching, ABO matching is important, and also matching of the HLA. When I was talking about polymorphism, I emphasized that it's very difficult to have a perfect match of the MHC, let alone other histocompatibility antigens. However, it has been found that there are certain matches are more crucial than the other one, and DR among them is at the top of the list. Okay, DR. Okay, so the DR is the most important. As you can see, the survival is the highest. And after that, the more the matches, the better they are. A haploidentical sibling is often a good donor if there's a DR match. Nowadays, with the immunosuppressive agents, it is not unknown that totally mismatched organs are also transplanted, particularly when there is a desperate situation. Okay, so there are some transplant clinics, and that I became aware only about a couple of months ago. Somebody from New York was giving a talk, and he said they often take unmatched donors. That, of course, does give the recipient time if they are in dire need for a transplant. However, it is ideally one should have a DR and a B locus match, or better than that is DR match with one haplo identity. Okay? What has been observed by empirical practice, and the mechanism for this effect is not really well understood or known, is that if the recipient is given small amounts of blood transfusion, 100 to 200 cc's, from the donor. Remember, they are matched for ABO, so there won't be transfusion reaction. That's most important, of course, to match that. But then after that, if the recipient of the organ receives 2-3 doses of 100 to 200 ml donor blood transfusion, donor type blood transfusion from the donor, the grafts work much better. So that has been sort of discovery from the practice itself, and it is practice. Okay? What the cause are, people have sort of hypothesized that there is induction of some sort of tolerance or hyporesponsiveness against the donor tissues. And that is the reason that the recipients, those who have received transfusion, okay, do better. This yellow line, okay? Now, all done, there will be not a hope for the organ to survive even with the best matches. Because not just MHC, there are other antigens as well, I told you about. So immunosuppressive agents are a must. Initially larger doses, and that is tailed off to maintenance doses. And what are the immunosuppressive agents? I've got a list of them here as well as on your handout. And what I expect you to do is remember the agents and what their mechanism of action is also. Okay? Most importantly, corticosteroids are used to reduce inflammation. They are anti-inflammatory. They alter T cell and PMN traffic. So by diverting them from the site of inflammation, it can reduce the chances of damage to the graft. Cyclosporin and tacrolimus, those are the two most popular immunosuppressive agents. And there's a third one I'll talk about that is in the same category. And these three work via the IL-2 pathway. Tacrolimus and cyclosporin, they inhibit the production of IL-2 cytokine. And remember, IL-2 cytokine is one of the most important ones that causes proliferation of the clones that are reactive clones. So they can inhibit production of IL-2 production by T cells in response to alloantigens or other antigens. IL-2, if you remember, Dr. McCallop told you this a few days ago, it also is needed for the differentiation of cytotoxic T cells. It also is needed for the activation of NK cells and a variety of other functions that this cytokine has. Okay. The third one is rapamycin, which is similar to tacrolimus and cyclosporin, but its mode of action is slightly different. It prevents activation of cells by IL-2. If you remember, IL-2 binds to this receptor, IL-2 receptor, and stimulates the cells, activates the cells to go into proliferation and also activation. Okay? So there's a very slight difference between the mechanism of action of these two and rapamycin. The first two, tacrolimus and cyclosporin, they suppress the signal induced by the antigen to synthesize IL-2, whereas the third one, rapamycin, it prevents activation signal by IL-2 via the IL-2 receptor. Okay? Now, the other few that are used is azathioprine, and it's a purine metabolism pathway it interferes with. Methotrexate, it interferes with folate metabolism. They're all immunosuppressive. Cyclophosphamide and melphalan, those are alkylating agents that can alkylate DNA, cross-link DNA, and therefore cells that are going into mitosis in response to antigen, like MLR in the case of transplantation, or activation by antigen, the DNA, if the DNA and RNA are cross-linked, obviously the cell is not going to go anywhere and it is going to die. Extremely toxic. There are other problems with the use of cyclophosphamide and melphalan, and their use is reducing now. However, they are still used in some transplant cases as well as autoimmune diseases. X-ray radiation is very, very non-specific, again, and immunosuppressive agent, and it causes damage to all the mitotic cells. Therefore, it would result in lymphopenia in terms of immune system, but also causes damage to other cells. And it is used only in the case of bone marrow transplantation. And because it is used to reduce the malignant cells, the leukemic cell burden in the host, in the recipient. In addition to that, there are some others listed in your handout. Anti-lymphocyte serum that is still being used. This use started in the 60s, and some centers are still using anti-lymphocyte serum. It's referred to as anti-lymphocyte serum, but it is a serum raised against T lymphocytes, and it reduces the number of T cells in a sort of non-specific way, and reduction in those T cells results in prolongation of graft. There are some other monoclonal antibodies that are much more specific to T cells, like anti-CD3 or anti-CD4 antibodies are also being used. There is an antibody that is often used in conjunction with cyclosporin, is anti-CD25. Now, CD25 is actually a part of the IL-2 receptor. So if you can eliminate cells that have IL-2 on them and which are IL-2 receptor on them, obviously that will dampen the immune response and prolong the graft. Okay? And those agents are listed in your handout. Okay. In the bone marrow graft system, what is the problem? It's not the acceptance of graft, right? Remember, where is bone marrow graft used? Either in leukemias or in immunodeficiency cases, where there's general immunodeficiency. So what will happen? Would the host reject the bone marrow if there's incompatibility? No. What would be the problem? Say it louder, please, so that... Okay, it won't make T cells. Any other?
The donor T cells would react to the host.
That's the most important reason. It is the complication. There is a complication. The human bone marrow has got lots of mature T cells. In addition to the precursors, it has all sorts of T cells, mature T cells. And those mature T cells, of course, will recognize the alloantigen. The CD4 cells will recognize class 2. CD8 cells will recognize class 1. They will proliferate, and the cytotoxic T cells would be activated, and they are going to cause damage to the host. And what is the name of that syndrome? Graft versus host disease, GVH. Okay, and you saw some complications like erythema, like gut mucosal cells are destroyed. Okay, so graft versus host disease is the most relevant complication. So it is important to monitor and remove those cells that are going to cause graft versus host disease. And how it can be done is by treating them either with antibodies against T cells and complement and lysing them, and then removing the dead cells, because dead cells themselves cause problems when they are infused into the patient. That's one way that used to be there. There's a neat way now that is being practiced, and that is removal of them by a magnetic system. And there is a machine that's based on magnetic system. And what one does is, in this case, I'll go over this, and then if there's a problem, I'll address that. One takes the donor lymphocytes, the donor bone marrow cells, that contains T cells as well as other cells, and assuming that the T cells are these little yellow crescent with the blue nucleus, the antibodies will bind to them. There are antibodies that are specific for T cells, and these antibodies are tagged with magnetic molecules, like iron. Okay? So these antibodies, as you can see, they've got a little asterisk there, and those are little magnetic particles, and they are chemically coupled to the antibodies, and when you mix it with the bone marrow cells, they will combine with the T cells, of course. And if that mixture of antibodies and T cell and cells is passed on to a magnet now, all the cells that have that antibody, meaning the T cells, will stick to the magnet, and what comes out of there is cells, bone marrow cells that are depleted of T cells. Okay? And that technology is being used for other purposes as well, research as well as clinical purposes, to remove a variety of cells against which you can raise antibodies, and those antibodies can be coupled to magnetic particles. Okay? So this is being used. In your book, in your POPs exercise, you will see something that there is an advantage in not removing all T cells. And what is done is that one removes most of the T cells, but spikes them with some T cells so they can stay there. And what is known is that in one particular case, this is practiced, is leukemias. In leukemias, it is of some advantage. By practice, it has been observed that it is an advantage to have a low-degree graft versus host-like reaction, and it is referred to as graft versus leukemia reaction. So you'll find that in your POPs exercise, and I'm just explaining it to you so that you know when you read it. Okay? So one can remove all of them, but it is advantageous in the case of leukemia to leave a few behind, or after removing to add back a few for graft versus leukemia effect. But if it were for treating an immunodeficiency, it is advantageous to remove all of it, because there you do not need any graft versus host reaction. Okay. Now, lastly, I told you that MHC is very important in certain diseases. Different people are susceptible or resistant to different infections to different degrees based on their MHC makeup. They've got a whole package of MHC antigens, obviously. 6 of the in a heterozygous, completely heterozygous individual, 6 of the polymorphic forms of class 1 antigens, and then another 6 to 8 or 10 polymorphic forms of class 2 MHC antigens. And those MHCs find one or other peptide to incorporate into them, and the more peptides they can incorporate, obviously the more resistant the person will be. So there is a genetic basis for resistance based on MHC. Also, certain MHC-bearing individuals are more at risk of certain autoimmune and inflammatory diseases. And the table in your handout just gives an example of a few of them. That does not mean that everybody who has got a B27 is going to come down with ankylosing spondylitis or Reiter syndrome. No. What it means is, given other circumstances, they're at a higher risk for developing Reiter syndrome or ankylosing spondylitis. And the same thing applies to other, okay? There is in the list you'll find in the book's longer list, and diabetes is included among them, and some other disease. Okay? That was all of for I had to say about MHC. I'll be happy to answer any questions while I change the